All right, well, as promised, I'm going to be recording the lectures for this last week. And this video for Monday, we're going to talk about individualized medicine and how our understanding of the human genome has shaped our ability to practice medicine and to treat some diseases. And so, as we talk through individualized medicine, we're going to emphasize a few elements of what we can do to make our treatment as focused as possible and not so much focused on just the particular pathologies. The first question we're going to deal with is this one. How has sequencing the genome helped improve and personalize medicine? And here we're focusing on personalize, but with that also comes an improvement of how we practice medicine. So the main way we do this is screening for particular mutations. Now, the mechanism we use for screening and the particular type of mutation may be different, but this is the primary means by which we begin personalizing medicine. So first part under here, we know that there are certain genetic illnesses that are caused by specific mutations. Now, many genetic illnesses have a suite of different types of mutations, but all in the same gene. So a good example of this is cystic fibrosis. The most common mutation in cystic fibrosis is a 3 base pair deletion that removes one of the amino acids of a little bit over 1,400 amino acid long protein. Now, that's the most common mutation in this gene is the CFTR gene, but it is not the only mutation that can actually cause cystic fibrosis. Several mutations in this gene could lead to an altered protein with the inability to function correctly. This is a chloride ion channel. Another example of a mutation that causes a genetic illness is sickle cell anemia. This is a mutation in one of the beta subunits of hemoglobin, and it leads to a misshaped hemoglobin molecule and a misshaped red blood cell. As a result, the hemoglobin is less able to absorb and carry oxygen, and the red blood cells take on a sickle shape. And the last one that we'll talk about in here is phenylketonuria, and this is the inability to metabolize phenylalanine. And when you metabolize phenylalanine, it converts it into another type of amino acid. And what happens here is this illness, this genetic illness, is linked to mental retardation. And so it is a specific mutation that causes this illness, and then that illness is linked with mental retardation. On top of mutations that cause genetic illnesses, there are also mutations that correlate significantly with genetic illnesses. Many types of cancer are this way, where it's not necessarily if you have this mutation, you will get this genetic illness. More if you have this mutation, you are significantly more likely to contract this genetic illness. And this is where we have a somewhat more interesting opportunity to personalize the medical treatment for these genetic illnesses. Many types of mental illnesses, we've talked about this throughout our semester, where you have copy number variants or translocations or inversions or even frame shift mutations that fall out of those categories that correlate with particular mental illnesses. And so right here, this is the CFTR, the cystic fibrosis transmembrane conductance receptor, and it is a chloride ion channel. And what it does is it functions to facilitate the diffusion of chloride ions either into cells or out of cells, most of the time out of cells, and water tends to follow the chloride ions. And so if you have, again, a suite of mutations can alter this protein, the most common of which is a 3 base pair deletion, resulting in one amino acid being missing. But regardless of the type of mutation, if the protein does not fold correctly, it cannot facilitate chloride ions diffusing out of the cell, and then water can't move outside of the cell. And so the most common pathologies associated with cystic fibrosis are really thick mucus buildups inside of the alveoli and the lungs, with the inability of water to move out of those cells lining the alveoli. The mucus becomes very thick, and so lung infections are incredibly common. This is one of the first pathologies noted with cystic fibrosis and tends to lead to the diagnosis early in childhood. Another pathology is the inability of digestive enzymes, some of the digestive enzymes, to get out of the pancreas. And so there are oftentimes digestive issues associated with cystic fibrosis. And then later in life, when the individual reaches sexual maturity, there's usually an inability to make gametes properly. And so sterility is another common pathology. So this is an example not only of a mutation that causes a genetic illness, but is also a good example of what you would call a pleiotropic gene, where you have a single gene that contributes to multiple different phenotypes. And here's our sickle cells. These are red blood cells, again, with abnormal hemoglobin. It's the result of a mutation in one of the beta subunits of the hemoglobin. Hemoglobin is a tetramer. It's a protein with quaternary structure made up of 4 subunits, 2 alpha subunits and 2 beta subunits. And a mutation, several different mutations in one of those beta subunits results in abnormal hemoglobin and then abnormal shaped red blood cells. And so you don't just have an issue of carrying oxygen, you also have an issue that these red blood cells with their sickle shape tend to clump and cluster inside of arteries. And so you can get significant blockages. But even just having sickle cell trait, where you have one normal allele for that beta subunit and one altered or mutated allele, is enough to lower the efficiency of red blood cells to carry oxygen and can present pathologies at high elevations. Here's the polypeptide that enables the conversion of phenylalanine into another amino acid. And there are several places on this polypeptide where a mutation can result in phenylketonuria. But in this box here, this is where you have the most common place for the mutation to happen. And this results again in a buildup of phenylalanine inside of the blood and correlates significantly with mental retardation. The average IQ of somebody with an untreated case of phenylketonuria is 55, which is fairly significant mental retardation. So as we start talking about screening, and again, all in the area of personalizing medicine, we need to address this question: how can we screen? And so, first place, and this is becoming more and more reasonable and more safe as time goes on, but we can, for many genetic illnesses, we can screen embryos for either malformed proteins or particular alleles. And this is especially easy in vitro fertilization, when you fertilize the ova outside of the female reproductive tract, and then you can, at the 8-cell stage, remove one of the cells and screen that cell for the malformed proteins or for particular alleles associated with malformed proteins. And because eukaryotic organisms, not all eukaryotic organisms, but many of them, and all deuterostomes have totipotent cells early in development, the 7 remaining cells will develop into all of the parts of the organism. We can also screen newborns for antibodies and for particular amino acids. And this is done with phenylketonuria almost every country on Earth screens newborns for phenylketonuria. And again, you're screening for a large number of phenylalanine amino acids in the blood. But you can also screen newborns for antibodies, and you can remove cells from newborns and screen their genome for particular alleles without necessarily having to sequence the entire genome. And then we can screen virtually anyone. We can screen children, we can screen adults for genetic illnesses, noting that many people are asymptomatic for genetic illnesses until they present oftentimes later in life. So we can screen anybody in a number of different ways, in the same way you can embryos and newborns, looking for malformed proteins, particular alleles, antibodies, particular amino acids. And then you can always sequence the genome. The cost of sequencing the genome decreases every year. In 2002, when the human genome sequence was completed, the cost of that genome was about $3 billion. The cost now is about $1,500, with projections that it'll be $100 or less in the next 10 years. And so this option is obviously going to be a lot more reasonable in the next few years than it is now, but is also a possibility for certain genetic illnesses, and if somebody is willing to pay that cost. Not necessarily something you can use for every individual, but certainly something that you can consider on a case-by-case basis. Now, something to keep in mind is while the genes may program or set something in motion, they don't guarantee the genetic illness. And this is true even for the illnesses that are caused by particular mutations, because we can alter the environment to reduce the chances of developing the genetic illness or even eliminate it altogether. And so while those illnesses may be caused by particular mutations, particular programming in the genes, we can alter the environment to overcome that. And so this is a really important point to keep in mind. And this is really oftentimes what we're doing when we are personalizing medicine. We are altering the environment to overcome what the programming is dictating. The next question we need to deal with is what makes for good screening? So yes, we can screen, and we can screen newborns, we can screen embryos, we can screen adults, but how do we develop good screens? And then the next question we'll deal with from this is what do we do with those data? And so first thing that makes for a good genetic screening is genetic screening should save money. And this seems to be an obvious statement, but is certainly something we need to keep in mind as we develop new methods of genetic screening. And right now, spending $1,500 to sequence the genome of every individual isn't a cost-saving mechanism. Therefore, insurance companies aren't going to pay for it. But there are genetic screens that do save enormous amounts of money, and insurance companies will pay for them. And phenylketonuria is a good example of this. The screening for this illness costs $2 per person for an illness that occurs in 1 in every 14,000 individuals. And so if you think about that, for every one person with this illness, we invest $28,000 to find that person with the illness. And while that seems like a very significant cost, if you think about that there's a really high correlation between this illness and mental retardation, the cost, $28,000 to find that individual, is a significantly lower cost than caring for somebody with an IQ of 55 for decades. And this is an example of where we have been able to personalize the approach for a person that gets a positive test for phenylketonuria. Another important one is that genetic screens should be repeatable. And so, if you think about something that's not a genetic screening, but is another medical screening technique, something like an X-ray or a CAT scan. And so what we mean by repeatable is if two random radiologists would look at the same test and come to the same conclusion, or thinking two pathologists would look at the same sample and come to the same conclusion, then that's a test that's repeatable. And those are successful tests because they're repeatable. A random pathologist, or a random radiologist should be able to look at one of those tests and come to the same conclusion any other pathologist or radiologist should. And so this should also be true of genetic screenings, that when you determine the presence or absence of a particular allele in a person, or even if you have access to the entire genome, any geneticist should be able to look at that and come to the same conclusion with regards to probabilities of developing certain genetic illnesses. Another thing is the screen should be accurate. So it's one thing to have two random geneticists come to the same conclusion. It's another thing to have those two random geneticists come to the correct conclusion. And so these screenings should be accurate, and there's really two parts of that. One is they need to have a high sensitivity. And so sensitivity is a measurement of the percentage of people with the illness that actually get a positive result on the screening. And so the higher the sensitivity, the lower the likelihood of a false negative, of actually having a negative reading for somebody that has the genetic illness. And then another one is high specificity. So specificity is a measurement of how successful is the screening at actually showing people that are truly negative that they get a negative result. So another way of looking at it is the higher the specificity, the lower the probability that you get a false positive. And so both of these are really important. Sensitivity is obviously it's a catastrophe to have a false negative for one of these genetic illnesses. Say to have a baby leave the hospital with a negative result for phenylketonuria, and it actually has that genetic illness and doesn't make the modifications necessary in order to prevent the mental retardation. The high specificity, getting a false positive is inconvenient and is a burden to be told you have a genetic illness only to find out later you don't, but doesn't carry the same weight. So we aim for genetic screens that have high sensitivity and high specificity, but sensitivity tends to be the one given a little bit more weight if you have to make a decision where you have to emphasize one over the other. And the last one is the screening should be safe, and especially considering that many of the genetic screenings are done on embryos or on newborns. Those are individuals that cannot consent to the medical treatment and have to have somebody make that decision on their behalf. And because of that, these treatments need to be especially non-invasive and safe for individuals that can't consent to those screenings themselves. And then so the last question we're going to deal with is this one. What do we do with the data? And we've talked a few times in class already, how do we make some of these decisions? And it becomes especially difficult to know what do we do with the results of these genetic screens. If you have a newborn and you determine that they have cystic fibrosis, what do you do with that? There's no cure for cystic fibrosis. There's no, as of right now, means by which we can change that CFTR protein. And so you have to kind of figure out, okay, well, what do we do with those data and how do we treat in the best way? Or even when you're trying to make a decision of do we screen, you should approach those questions approach those questions with these same goals in mind, and that's to make sure that whatever decision we make, we consider it to be a morally good decision based on our ethical framework. And so we've talked before about the need to really combine several different areas of ethical thinking. Divine command theory, asking this question, has God revealed his desires? And so this isn't necessarily just God commanding us to do something or forbidding us from doing it, but is God just revealed in his word his desires on how we ought to operate among these types of people, or to make a decision in this setting. And a big one here, and something that I think is heavily should heavily influence our thinking here, is this idea that James talks about this in his epistle, that true religion is ministering to orphans and widows, and you get this sense that there's this real need and this real image of what Christianity truly is, and that's serving those who are weak and serving those who in many ways can't serve themselves and can't make decisions for themselves. And I think that ought to influence how we operate and think and make decisions for embryos and for newborns. Another question is, how can we enhance the image of God in people? And this really should be all people, treating every person as an image bearer, because that's the reality of Scripture, that every individual is created in the image of God, and because of that has worth. And this is oftentimes forgotten in utilitarian thinking, where we try to maximize the good for the most number of people, forgetting that every single individual is important because they're made in the image of God. And when we talked about virtue ethics in class, I talked about just having a moral exemplar that you could model your life after. And that's a really important feature of virtue ethics. But there are specific virtues as well. Aristotle talks about 4 virtues: wisdom, temperance, fortitude, and justice. And then Aquinas, I think it was, added faith, hope, and love as Christian virtues, and kept Aristotle's 4 virtues as well. And so it's really difficult to find somebody, even in our lives, that would be an exemplar for each of those virtues. But we could find, some of those virtues. So what Christian virtues influence our decisions? We can find a particular virtue that influences how we ought to make that decision, and then find an exemplar of that virtue and model our decisions off of that individual. And so this becomes especially helpful as we're making some of these decisions on what do we do with these screens. How do we enhance the image of God in this person, in this embryo, in this newborn, even in this adult that has this positive result on some genetic screen? And what are Christian virtues, especially love and justice, I think influence these decisions a great deal, and even wisdom. How do we make these decisions that are going to impact the rest of people's lives? And then really practically, we use these data to treat people, and to treat the problem and not the symptoms. And this is where medicine gets very individualized, and the ability to screen for particular mutations lets us know not only the cause of the particular pathology, but even the cause for the cause. And so that we can begin to treat the underlying issue and not just manage the symptoms. And so obviously, we've talked a little bit about gene therapy, and that's what we're going to focus in on Wednesday. That's why I put this one as the first on our list. We're going to look in at gene therapy and how do we help to reprogram people. I mentioned earlier that the genes program, they don't necessarily guarantee the expression, but they do present the programming. And so we'll look on Wednesday, well, how do we work towards reprogramming individuals to where we're not just managing the environment, but we're also helping to redo the programming so that in most environments, the person is going to operate normally, operate the way they were designed to operate. Hormone therapy is a very powerful one for many of these genetic illnesses, if you understand the underlying cause of the pathology, you can try to override that by changing the environment. And this is where hormone therapy can be especially successful. And you see it used a lot with alterations in sex chromosomes. And we saw this earlier in our semester, where let's say, for example, somebody with Klinefelter syndrome, that's XXY. So they have a Y chromosome, therefore they are male, but they also have two X chromosomes. And so they have some unusual characteristics. Well, understanding that and having the understanding of what that means, we can actually treat that extremely successfully by altering the environment using hormone therapy. And diet is an especially powerful way to treat a number of genetic illnesses, those that just correlate significantly with mutations, and even some of those that are caused by mutations. So I've mentioned several times in this set phenylketonuria, and it's the inability to metabolize, break down phenylalanine, and so you end up generating large amounts of phenylalanine in the blood. And a way that you can treat that in a very successful way is just to implement diet changes. Feed individuals with phenylketonuria diets extremely low in phenylalanine, so you don't get that buildup, and is actually a really successful way to almost eliminate the probability of developing that mental retardation. So I mentioned before, the average IQ of somebody with an untreated case of phenylketonuria is 55. The average IQ of somebody with phenylketonuria that's treated with a diet low in phenylalanine is average. It's the average IQ. So what do we do with the data? Well, I think we need to be very careful and thoughtful in how we do this and make decisions that are consistent with our ethical framework and something that we can consider morally good, keeping in mind that there aren't any amoral decisions. They're just, decisions that there may be decisions that are morally neutral, but oftentimes not. There are decisions that have some moral good, and we can help ourselves to make those decisions by asking the three questions: has God revealed his desires? How can we enhance the image of God in every individual? And what Christian virtues can influence this decision? And then from there, very practically, we can implement specific changes to the environment with hormone therapy and diet therapy, or even changes to the programming with gene therapy. So that's it, and we'll see you on Monday, and I'll post a video for Wednesday on gene therapy.